Okay, there we go. Much better. Okay, welcome everybody to the 2020 or 2026, the start of Cafe Scientifique. My name is Jennifer Cobb. I am the chair of Biochemistry and Microbiology in the Faculty of Science at the University of Victoria. The Faculty of Science is sponsoring this event, and it's a real pleasure to introduce tonight a colleague, Dr. Brad Nelson. So Brad got his bachelor's degree at UBC. He then went on to UC Berkeley to do a PhD, and then he was at the Hutch, the Fred Hutchinson Cancer Institute in Seattle for a postdoctoral fellowship. After that, he then became the founding director of the Dealey Research Center, and he is now the scientific director of the immunotherapy program with BC Cancer. He is joint appointed at both UBC in medical genetics and in biochemistry and microbiology at UVic. This fall, he was also inducted as a fellow into the Royal Society of Canada, and that's a huge honor right here locally. So his lab is really pushing forward both on the research front and in clinical therapy, immunotherapy. And his lab is working both on the research side, but he's also done an enormous amount of work to get immunotherapy into clinical trials. And the work that he is doing right here at Royal Jubilee Hospital is really amazing. And I hope we'll get to learn some about that tonight. He's also, I learned this over the weekend, he's somewhat of a child prodigy guitarist. And he said he's been working his way through the Baroque era with forays into country, jazz, and pop. So the title of his talk tonight is Engineering the Immune Response to Cancer, Local Efforts in a Global Context. Please join me in welcoming Dr. Brad Nelson.
Well, thanks, Jen, for that warm introduction. I don't think I'm a child prodigy, just I've been plugging away since childhood. And thank you to all of you for coming on a cold Tuesday night. At least there's been a pause in the atmospheric rivers for a day or two, so I'm appreciative of that. And tonight we're going to talk about cancer immunotherapy field that I work in. And I don't have the benefit of PowerPoint slides, so I feel, this is going to be a bit of a different presentation for me. So I'm just going to talk as if the slides are there, but if I start pointing at the wall, you'll know why. And I thought the way I would talk about this, so cancer immunotherapy probably many of you have heard about this. This is a revolution in oncology that's happened, just seemingly come out of nowhere, I think, to a lot of people in the last 10 or 15 years. But like all breakthroughs in science, it's actually decades in the making, in fact, generations in the making. And I'm old enough now that I've been around for one of those generations. And so what I thought I would do to help introduce you to the topic and also give you a sense of how science works and how progress is made is just tell you my story of how I got into cancer immunotherapy and ended up where we are today. So we're going to start back in the 1980s. Actually, I did graduate from UBC, but I started at Camosun and I went to UVic along the way. So I've actually been to all three of those schools. And was very interested in neurosciences back in those days, and ended up going to UC Berkeley, as you mentioned, to get my PhD in neurosciences program. And somehow I got distracted even there and started working on developmental biology, or embryology, and was actually studying leech embryos, trying to understand how a leech egg turns into a leech. So very, very basic research. It was fun to do at the time, very interesting. We had some really cool results, but I was constantly being asked, what the hell are you doing? Why would anyone care, how a leech embryo develops? I guess that's a fair question. It's a lot you can learn from that, but that was a question I was constantly getting. Toward the end of my PhD, and many of you would know when you finish a PhD, the next step is typically postdoctoral studies, and then on to, if you're lucky, a faculty position somewhere. So I was just winding up my PhD and thinking about what do I want to do as a postdoc. And then very sadly, at that time, my mother-in-law was diagnosed with ovarian cancer. She lived here in Victoria. And that was a real shock to our entire family, and of course to her in a major way. And it was the first time we'd really come face to face with cancer. I hadn't thought much about it up until that point. And this was the late 80s, and the treatments that were being offered at that time, so the world of oncology was still very much hooked on chemotherapy as the main treatment. Radiation therapy as well, I should say, but for ovarian cancer, radiation doesn't work, but chemotherapy had some efficacy. And you may or may not know, chemotherapy came about in the 40s and 50s and really had its heyday through the 50s, 60s, 70s, 80s, and continue to be advances today. But in the late 80s, it was still the thing. And what was being tried was giving really ultra high dose chemotherapy. And the limitation of that was that would wipe out the patient's immune system and their blood compartment. So they would give the high dose chemo, but then give the patient a bone marrow transplant, their own bone marrow, so an autologous transplant to rescue the blood compartment. So that was the clinical trial she was offered in Vancouver at the time. And knowing that she had a very serious disease, she signed up to that, for that with everybody's encouragement. And unfortunately, underwent the treatment and ended up, the toxicity from that was so high, she ended up in a coma as a result of that. So just an absolute, terrible outcome for her and for all of us. And so here I was thinking, I was absolutely shocked that this is the state of cancer care in what at the time felt like the modern era. I thought it's just shocking to me that her cancer wasn't detected earlier or couldn't even be prevented, and that these are the treatments, this is the best we could do. She eventually came out of the coma after a couple of months and started to bounce back cognitively, but then the cancer came back just around the same time. And so she then signed up for another clinical trial. So Taxol was a new wonder drug chemotherapy agent at the time, and is in common use now, but it was pretty brand new at that time. So she signed up for a trial at the University of Washington for Taxol, and was able to get on the trial, received that, but that kind of wiped out her blood cell compartment. And as a result of that, she actually became septic and ultimately succumbed to her disease and to the treatment in general. So again, just a very sad, tragic story and very inspiring story for me and many others, but just thinking, okay, I think I know what I want to do now. As a postdoc, I want to get involved in cancer research. And meanwhile at Berkeley had been building up its immunology program. So they'd had a number of what we call job talks, so candidates coming through giving talks about immunology, and I've been going to those. And so I've been learning a lot about the immune system just by going to these seminars, and I just kind of instinctively thought the immune system has to have something to do with the solution here. This whole idea of immunotherapy back then was pretty fringe. It's an idea that's been around since the early 1900s. It wasn't a new idea, but it had never really worked. So it's one of these great ideas that doesn't work. But still, I had just an interest in it and a gut instinct that this is going to be part of the future of oncology. And I don't say that like I have some great foresight. I really went in kind of naively also, but as we'll see, turned out to be a good hunch. So I was lucky to get a postdoctoral appointment at the Fred Hutchinson Cancer Research Center in Seattle with one of the world leaders in immunotherapy at the time, and still a leader, Phil Greenberg. And he was just an absolutely fascinating character to me. I've been used to sort of hippy-dippy professors at Berkeley in my embryology group, and loved them, but Phil was just the opposite, clinician scientist. He had wild, long, white hair and drove a sports car and just was fast thinking, fast talking from Brooklyn, and just his energy was incredible. So I was excited to sign up to join his lab. And they were, this is the early 90s now, they were, he was the first person to show that you could cure leukemia in mice using T cells, immune cells. So that's, that was his thing. That's what they were working on. But in the early 90s, of course, AIDS was a huge problem. And so he had pivoted for a few years, part of his lab at least, to study AIDS. And the same idea, could we cure AIDS by giving people infusions of T cells? And great idea. A challenge there is, of course, AIDS, the HIV virus actually infects T cells, as you may be aware. And so, how do you treat infected T cells with more T cells was one of the challenges. The project I was given was to genetically engineer the T cells. The idea here is, okay, so now I'll introduce you a bit to T cells. These are our immune cells. We've all got them circulating in our blood. They're part of our white blood cell compartment. And they're the cells that go around and they monitor the body looking for diseased cells. And there's, at a first approximation, there's like two types of T cells. There's the killer T cells and the helper T cells. And so the killer T cells are actually licensed to kill. They're able to go up to a diseased cell, and if they decide it's a diseased cell, they can kill it. They actually glom onto it, like stick to it, poke holes in the cell membrane, inject toxic enzymes, and it can cause that cell to just dissolve within minutes of contact. So they're very, very powerful. You don't want to piss off the killer T cell. They can also cause autoimmunity. So there's all sorts of checks and balances to keep them in control. But they are absolutely essential for us to all remain healthy against infections as well as cancer, as we'll see. The other type of T cell are called helper T cells, and they make growth factors and other factors that help the killer T cells do their job. And it's a bit like a missile silo where you need two people to turn the keys at the same time in order to launch a missile. In the immune system, both the helper T cells and the killer T cells need to agree that this is a diseased cell, and if that's the case, the killer T cell becomes licensed by the helper T cell. And the problem in the AIDS setting is that the helper T cells are infected by the virus, and AIDS patients lack functional helper T cells, and so the killer T cells weren't doing very well on their own. So my postdoctoral project was, can we genetically engineer the killer T cells so that they don't need the helper T cells anymore in the setting of HIV, and if so, and then we could apply it to cancer. So I got onto that project. Won't get too into the details of it, but really forced me to learn, and this is kind of first day on the job. I've just been playing with leeches the day before, and now I'm genetically engineering T cells. And it's 1991. We didn't have a lot of great tools to even be doing these things, but we took a good stab at it, and I came up with an engineering solution that after a couple of years showed that it was really working, and actually was sort of my career making some discoveries at that time. Really made my career, really cemented me as an independent researcher, and Phil ended up offering me a faculty position at Fred Hutch, and my career was sort of launched as a result of this work. Meantime, AIDS got dealt with in another way, which is the antiretroviral drugs came along, and so the whole approach of T cell therapy never really was needed, got replaced by something even better and simpler. But nonetheless, we had really sharpened our axe during this time. Phil's lab had now learned how to make T cells for clinical use. We learned a bit about how to genetically engineer them, and the stage was really set now to go and apply these ideas back to cancer, which is what we were all primarily interested in. So that was a number of years in Seattle working in that area. At this time in the cancer field, chemotherapy was still a very promising and very widely used approach for sure, as it is today. But what had really come along was genomics and the whole idea of designer drugs, silver bullet type drugs. Now we knew how to sequence the genome, we the world, and so we could sequence people's tumors and we could find out what are the oncogenes that are driving that cancer, and let's just make really smart drugs that switch off those oncogenes and we'll be done. That'll take care of the problem. And so for the 90s, I think can be characterized by genomics and these coming up with these real designer type silver bullet therapies. And there were some great successes that came out of that, like Herceptin for breast cancer would be an example. Prostate cancer medications, androgen inhibitors are another example. Gleevec for type of leukemia was another example. But for most cancers, this approach proved to not work so well. It made drugs that switched off oncogenes, but it became what is commonly said in the field a whack-a-mole type problem where, yeah, you can kill many of the tumor cells with this silver bullet drug, but tumors are constantly evolving, much like viruses do. And so you just get the cancer coming back, and now it's found another oncogene that it likes just as much. And so your original oncogene is irrelevant now, and the drug doesn't work. So people would have these remarkable responses, but then the cancer would come back, and that same drug was now essentially useless. So this takes us to about the year 2000 or so. But nonetheless, genomics was, really a big thing. And I personally was no longer thinking immunotherapy for cancer necessarily made sense, given that, like many people, I thought, well, it's just a matter of finding a bunch of oncogenes and having a medicine cabinet full of designer drugs, and we'll just, be able to give everybody the right cocktail of drugs, and we'll be done. And we'll figure out this whack-a-mole thing just by brute force, if you will. I was then, 2003, offered just an amazing opportunity to come back to BC from Seattle to head up the DMD Research Center here in Victoria, which is part of BC Cancer. And it was a brand new research center that the community had built. And It was a blank canvas, and I was offered the position of founding director and a couple $1 million to get the place started up and to hire some staff, et cetera. So a fantastic opportunity. And I'm eternally grateful to this community for building this and making this possible. And they said, so what do you work on, of course, as part of the interview? And I said, immunotherapy, which at the time was even fringier, perhaps, than it had been even in the 80s. I think they were hoping I would say genomics, but no, immunotherapy. And they said, but the VP research who hired me, Victor Ling, was a believer in playing the long game and really thought there was a place. So he was actually quite excited that we can get somebody in Victoria to run an immunotherapy program. And so I was hired to do that and got going with that. One of the recruitment committee, Ivo Olivato, wouldn't mind me saying this, but he said to me after I was hired, he said, you know, I voted for you because you seemed like a nice person who would be a competent director, but I don't believe in immunotherapy. And I said, fair enough, you know, why should you believe in something that doesn't work yet? But you know, give it some time. Let's just see what we can make of it. So got to work with this, and I'll tell you the day that I started to really believe immunotherapy could work was we had some mouse experiments we had set up, and we went to a lot of trouble to make a mouse model of breast cancer that we could treat with T cells, and spent years actually just building this model and breeding the mice and getting it all ready, and we did our very first experiment. So we had a mouse with multiple tumors actually, and gave some T cells. We weren't actually expecting to have much of a therapeutic effect. We were actually assuming the tumor is going to shut down the T cells, but that would give us a chance to figure out how did that happen, how did they get shut down, and how can we reverse that. But I was in Seattle, I happened to go back to the Hutch that day and called up to Victoria and say, how is the experiment going? And Erica Wall, who was working in my lab at the time, said, you won't believe it, the tumor's gone. And this was a large, spontaneous mammary tumor. And not a huge dose of T cells. And she said, I've checked and double checked, it's the right mouse. And so that mouse we called special because she was cured of her tumor, and we all just let, jaw-dropping sort of result that obviously I remember to this day. So kept plugging away at this and started, thinking about someday what would this look like in terms of clinical trials. Meanwhile, I'll just check my time here. Okay. What had happened in the rest of the world? Well, we started hearing about, so now we're maybe approaching 2010, started hearing about some amazing clinical results with this class of immunotherapy drugs called checkpoint inhibitors, or immune checkpoint inhibitors. There's a few different names for them. So if you remember, I said that back at Berkeley in the 80s, they'd been building their immunology department. Well, one person they hired was Dr. James Allison, who was a basic immunologist, looking at really fundamental immunology. And I'll tell you a bit more about T cells. Is like all cells in our body, cells communicate with each other all the time. They have to in order to form a functional body. And so cells have receptors on them, like little antennas on the surface that they use to send and receive signals to each other. And so T cells, in order for them, especially killer T cells, to become licensed to kill, there's actually a number of receptors that need to be triggered in order for that full licensing to happen. So Jim Allison had been studying one of these receptors, which he initially thought was a receptor that turned on the T cells, stimulated them. But the more experiments he did, he realized, no, actually this is inhibiting the T cells. It's like a braking mechanism on the T cells. So it's a way of keeping the T cells under control. And when they started looking at tumors, they saw that this braking mechanism was flared up, very active on the T cells in the tumors, and sort of realized, wow, these T cells are, somebody's pumping the brakes on them and stopping them from attacking the tumor. Not somebody, the tumor is doing this. So he had the simple but brilliant idea of, let's make an antibody that would block that braking mechanism. So break the brakes, if you will. And so now you can have your foot on the gas, take your foot off the brake, and the car goes forward, right? And so they started doing experiments in mice, getting great results. Pharma companies got involved, made clinical grade drugs that did this. And the first one that came out was a drug called ipilimumab, and they first tried it in patients with melanoma and started getting just astounding results. So the drug is just an antibody, so just a simple protein that blocks this braking mechanism on the T cells. And give that to patients, and the immune system is unleashed. And some patients can have a complete response, complete regression of their tumor as a result of this. Not everybody, but a good solid 25 to 30% of patients would show a really good response. Others, meanwhile, Dr. Hanjo in Japan discovered the different pathway, the PD-1 pathway, and same idea there, another braking mechanism. T cells actually have probably a dozen or so different braking mechanisms. It's quite complicated. But another drug was developed, or class of drugs that targeted this other brake called PD-1. And that has proven to be even more effective than the first class. And so there's drugs, common names are like pembrolizumab and nivolumab, for those who may have heard these names. So those drugs, this class of drugs, has just swept oncology now. So now we're into early, let's say 2010 to 2015. Almost every week there was a new FDA approval for these drugs being approved for, initially for melanoma, then lung cancer, cervical cancer, lymphoma, breast cancer, you name it. Not every cancer, but many of the big ones. So in the dozen or so years I had my new job in Victoria, immunotherapy had gone from being this fringe thing that Ivo Olivado didn't believe in to something where everybody in the building is getting these drugs now, practically. And Ivo did come back to me around that time and said, you know what, I was wrong. I think there is something to this immunotherapy. Now, I want to be clear, I'm not taking any credit for this. This is something that happened globally, and maybe my research, had a small contribution to our understanding that led to this, but it was really a number of groups around the world that made this happen. So we've got special being cured of her tumor. We've got these checkpoint drugs coming along. Let's check back with ovarian cancer. Problem there is these checkpoint inhibitors do not work for ovarian cancer or for quite a few other cancers that we all care about, pancreatic cancer being another example, and many of the blood cancers. The drugs might have a little bit of an impact, but not enough that to really benefit the patient. So for ovarian cancer, there still is no approved immunotherapy. So there's still work to be done, obviously. But along came another breakthrough, completely independent, around the same time. And this harkens back to the kind of work we were doing in Seattle with T cell therapy. And so this is the therapy I'm going to talk about is CAR T cells. I guess I'll just ask, how many people have heard of CAR T cells? Quite a few. Yeah. If I'd asked that 5 years ago, probably no hands, but more and more people are hearing about it. So we'll talk about that next. So let's just start with what are CAR T cells. So CAR T cells are genetically engineered T cells. So you have a cancer patient, you get a blood sample from them, you isolate their T cells from their blood, their helper and their killer T cells. And we put a gene into them, an artificial gene called CAR, which stands for chimeric antigen receptor. And the details aren't worth going into without my PowerPoint, but basically these CARs are a hybrid, a chimera between an antibody and a T cell receptor. And so what they do is they kind of hardwire the T cell to be able to recognize the tumor cell. And depending on which antibody you use, you can target the T cells to any cancer you want, essentially. Where they have first been used successfully is targeting leukemias and lymphomas. And they target an antigen called CD19, which is on those tumors. Again, the details aren't super important, but just imagine the tumor has a flag on it, and we can make the CAR T cells that recognize that flag, and they will go and kill any cell that has that flag on it. So this is work. So around 2011, the first paper came out in the New England Journal of Medicine. Just 3 patients treated with CAR T cells, pediatric patients with leukemia, and they had complete responses, complete regression of their tumors. These are kids who, the fact that they were on this trial meant that every other therapy had failed them to this point. This was a highly experimental therapy, and yet absolutely remarkably seeing these complete responses. So this just shook the oncology world in a good way, like, wow, genetically engineered T cells can actually cure cancer. Absolute game changer. Where had that therapy come from? Well, it had actually started back in the 80s. An Israeli group had come up with the original concept. It was during the 1990s that people figured out how to engineer T cells. That's the kind of stuff I was doing back in Seattle. And actually, the HIV virus proved to be a really good vector for delivering genes into T cells because it infects T cells so well. You could strip it of its bad genes and put the CAR or other genes in there, and it would deliver them into the T cells. So through the 90s, people were figuring out how to do all this, and then the early 2000s, the clinical trials were happening, and the reports started coming out around 2010, 2011. Absolutely shook oncology once again. So we've got the checkpoint inhibitors, we've got CAR T cells, and suddenly immunotherapy now is like the hottest thing in oncology. And people in Vancouver and across the country are saying, why is our immunotherapy program in Victoria? You know, it's like, well, hi. You know, from the beginning, I had the privilege and the good luck to be here to be able to build this program. Okay, but we're still working in mice, and now kids are being cured of leukemia. You know, how do we really get in the game here? So 2014, I got called into Doug Nelson's office. He's the president of the BC Cancer Foundation at the time. And I didn't know what I had done to get called into the office, but I went, and he said, you know, we're thinking of a big campaign. If you could do something really big, what would it be? And I said, well, T-cell therapy. But it's really expensive. We're going to need a clean room. We're going to need specialized staff. Nobody knows Canada knows how to make these. And he said, well, how much? Let's give me some numbers. So about after an hour, I walked out of there with a $4 million commitment from the BC Cancer Foundation. Another thing I'll be eternally grateful for, to start hiring a team and building a clean room in Victoria to start making, we ended up making CAR T cells. So I was then able, my whole team, we were able to leverage this with some federal funding, some provincial funding, put together, more like a $15 million package in total. And we were able to actually build the clean room and hire people and figure out how to make CAR T cells. And within a few years, we were able to open our first clinical trial And we did this in collaboration with folks in Ottawa, because our vision has been to produce a made in Canada CAR T-cell product that's affordable and accessible within our public healthcare system. So the part I forgot to mention is CAR T-cells, the commercial version of this is very, very expensive. Very effective, but the commercial products, there's three for CD19, and they range in, the approximate price is about $500,000 per patient just to make the CAR T-cells, and then you have to add the clinical cost to that. And so in the US, this is like $1 million cancer treatment, but very effective. So, the cost benefit, makes sense, at least from some perspectives. But with just to give some perspective, for oncology drugs, we're kind of used to spending about $50,000 per patient on sort of your average run-of-the-mill cancer treatment. Those are the kind of numbers that our system can afford. The really expensive designer drugs might be $150,000 in that range. But to be talking north of $500,000 is a lot of money, and it's affordable as long as it's just some leukemias and lymphomas. But the day we figured out how to get CAR T-cells working for other cancers, this would become rapidly very unaffordable. Our center in Victoria, we see 5,000 new cancer patients a year. If every one of them needed a $500,000 cancer treatment, that would be billions of dollars just for our center in Victoria. So just to give you some perspective on that. So my colleagues and I across Canada, we came together and said, between our different groups, and this was actually at a pub in Halifax after a conference, we had this conversation that my lab can make T-cells, John Bell's lab can make the lentiviral vector that's needed for them, Rod Holt's group in Vancouver can design a CAR. So we put all of our expertise together and came up with this made in Canada CAR T-cell and opened our clinical trial in 2019. There were a lot of doubters. Many of our colleagues across Canada were like, well, you guys aren't going to get blown out of the water by commercial product. You won't even get to treat your first patient before the commercial products are here, despite the cost. And we thought, well, hope that's not true. Commercial's not here yet, so let's just keep moving forward. Then, of course, COVID came along, and suddenly supply chains are disrupted. Patients were being sent down to Seattle for treatment. That was now very difficult. And our trial was open, and so we started accruing a lot of patients very rapidly. And same story with our colleagues in Ottawa. So quite quickly, we finished our phase 1 and moved into phase 2, and we've now treated 95 patients across the country now. And I'm pleased to say, proud to say, we're getting results that are, it's not a head-to-head comparison directly, but would appear to be equivalent to the commercial product, but at about 20% of the cost, and much faster turnaround time as well, which is better for patients as well. So, thank you. And I'll also say of the cost, so it's about $125,000 per patient for us to make the CAR T-cells. Most of that money, over half of it is jobs. That's salaries that we're paying for creating the next generation of CAR T-cell manufacturers, a job description that didn't used to exist, but now does, as well as the quality control people, the quality assurance, the regulatory people. So really creating a new generation of people who their starting point in life, whereas mine a generation ago was, quite different. How do we even get a gene into a T-cell? Now for these young folks, their starting point in life is, oh sure, we can genetically engineer T-cells, and we can use CRISPR now, and we can actually, just one gene, that's boring, we can edit 10 different genes at once if we want to. So their minds are completely open to the idea that anything is possible in terms of engineering immune response. So, that just shows what can happen over a generation of research. So we're at the time, and I'll just wrap up though with where are we going from here. So with this first product that I mentioned, treated 95 patients, we're now talking to Health Canada about getting what's called market approval for this. And our hope is that we could get market approval so that we could actually produce this and actually get reimbursed to make it by Ministry of Health, which would then, of course, create a revenue stream for us to help pay for our operations, while saving the healthcare system money and creating jobs and all the good things I talked about. So we're hoping to really complete the full loop there by taking it to that level. Happy to talk about what are the challenges there, and one of them is commercial product is here now, and so we are in competition with commercial product from that regard, and that does pose some problems, but nonetheless we're charging ahead. So that's one thing. The other thing is, okay, how do we then, let's get back to ovarian cancer and other cancers like that. So far, nobody in the world yet has come up with a good CAR T-cell strategy for treating solid tumors. And by that I mean non-blood cancers. So if we talk breast cancer, lung cancer, pancreatic cancer, ovarian cancer, CAR T-cells don't work yet in those settings. They do in mice, but not yet in humans. Why is that? Well, the CARs can work perfectly fine just from a molecular perspective. It's not that. It's that these tumors, these solid tumors, are tough for the T cells to get into, to really penetrate into, to do their killing activity. And you could just imagine a piece of bristly meat that's what tumors often look like. They're just full of sinew, connective tissue, and they have limited blood supplies, and they're a mess. Tumors are not well organized structures at all, and so it's hard for the T cells to get in and do their job. There's other barriers as well, but that's so breaking the solid tumor barrier is a big goal of the field. And so we are charging ahead, and we've got a CAR T cell now against a target called mesothelin, which is on ovarian, pancreatic, and endometrial cancers, as well as a few other tumors. We're getting fantastic results in mice at this point, far outperforming CARs that others have used in this setting. So we think we've got an exceptionally good CAR. We think that's not going to be enough on its own, so we're also building a throttling mechanism that we can put into the CARs, so that if the T cells start petering out, not trying hard enough, we can step on the gas with them through this engineered receptor, which actually is inspired by work I did back in the 90s as a postdoc, but we've got a 2026 version of this that's working beautifully in mice. So we want to put those together and hopefully come up with a really potent cell therapy, T-cell therapy that now has clinician control as an added feature with this, so that we can titrate the CAR T-cell activity to just the right level for each patient in real time. And I'm pleased to say that our BC Cancer Foundation continues to fund us generously, and they've got an ongoing immunotherapy campaign right now. I'm not here to raise money, but just in case you're interested, we have an active campaign because we want to get these new innovations into the clinic, and we're hoping that our ovarian cancer CAR will enter the clinic about a year from now, 2027. So to summarize, I think most days it feels like cancer research moves really slowly, in that you're always waiting for that next result or whatever. Science in general, on a day-to-day basis, can feel it's a hard grind a lot of the time, and yet when you look back at what the world can accomplish, I've just told you a 35-year story, it's really pretty remarkable the progress that can be made. So there's lots of work more to be done, but I'm very faithful, I have great faith in the idea that immunotherapy is going to be an even bigger part of cancer treatment in the future. And I'm also inspired by other types of therapy, non-immunotherapies. There's huge progress being made there as well. So what we're doing is, as a research community, is working, and we just need to stick at it. So I'll stop there and happy to take any questions. Thank you.
Could I ask you?
Yes.
You were saying that the commercial aspect had come and taken your product and was flaunting it or selling it or whatever. I don't understand that, because wouldn't you have prioritized your research?
Yeah, so I alluded to, there's a competition with commercial products
Yes.
now, and I didn't mean to say that it's not that they have taken our product. Rather, we've got a competing product, and the issue we face is patients now have access to the commercial product, so it's unethical to offer them our experimental product when there's a commercial Health Canada approved alternative. And that's the way with any drug, and I fully believe that's the way it needs to be. I'm not complaining about that, but that's, so our job then is to actually prove that we are not only equivalent to commercial, but that we're better, that we have advantages over commercial, so that there's now a reason to prescribe our CAR T cells over commercial. When Health Canada looks at things, I should say, they don't take cost into consideration. They, their primary, their only concern is that of the patient, what's best for the patient, and the system just needs to work out the cost. So, although everyone can see that we've got a cost advantage, that we have to also show it's a superior product.
How did they get your info?
Well, we presented to them. So we
Oh, you did?
Yeah, to Health Canada, I'm talking about. Yeah, oh, absolutely, yeah. So we go and well, they give us approval to do the trial, and then we come back and we say, we did the trial, here's the results.
Oh, and then
Can we
they
have
take it
market
up.
authorization, please? And
Oh.
they say, well,
God, that doesn't seem fair.
Okay, a few other questions. So for the advantage of the people that are online, if you say the question, I'm going to repeat it so that they can hear that before the answer. Okay, back there. Yeah, you.
Hi, a couple of days ago, I just read in the Cell magazine about Dr. Zantra at BBC, who's been able to develop stem cell to help make helper T cells in a viable way that's not arduous and very expensive. Like, it looks like it may be a potential for commercial use. What do you think that would do for your solid tumors with your CAR T cells, killer cells?
yeah, so you're referring to, and this is Dr. Peter Zanstra at UBC, who's a colleague and a friend of mine as well. And so the general idea there is, can we make big batches of CAR T cells that can be given to multiple patients? What I didn't say today is, or didn't say directly at least, is we make CAR T cells, they are handcrafted for each patient. We start with that patient's blood sample and we engineer their T cells and give them back their T cells. Could we save money by making a big batch of CAR T cells that could be given to 100 patients? And the challenge there is that it's like organ transplants, right? It's just, you get rejection of T cells. So but people like Dr. Zanstra are working on that problem, and the challenge there is, can we make the CAR T cells stealth enough that they don't get rejected by patients? So they have to do some more gene engineering to those in order to make that happen. And so in his case, they're able to start with stem cells and coax them into becoming T cells, and then they can edit them to make them stealth T cells, and the hope is then to be able to treat multiple patients.
So if they start from stem cells, will they still provide that rejection if you develop them as?
Yeah, the fact that they're from stem cells alone doesn't get you past that problem.
Doesn't get you past.
No.
Thank you.
Given economies of scale and AI and other developments that are likely to happen over the next decade, what would your best guess be of what the price per treatment might drop to for the year, say 2040, 2050, for the bloodborne cancers that CAR-T currently works for?
Yeah, so with, just to repeat the question, with, with advances in various technologies, what could the price look like in 40 or 50 years? I think it's pretty clear there that we won't be making the CAR-T cells in the lab anymore, but we'll be injecting the vector directly into the patients and letting it actually affect the T cells in the patient's body and produce CAR-T cells that way. So that's called in vivo transduction. And there are a number of companies working on this and even doing clinical trials of that idea now. There's a lot of issues, a lot of challenges with it, but it does hold promise for the future. So I think that's where we'll be. We'll look back at our clean room that we built in Victoria, and it'll be like, the computer rooms of the 1970s that, now your iPhone has more power than that. But still, we need the clean rooms now in order to get there. Right?
In the back.
I'm actually currently going through immunotherapy and chemo right now, and I was just wondering why it's like a once every 3 week thing rather than a weekly thing. I guess I could have asked an oncologist, but I forgot. I have chemo brain so bad right now, so I'm just curious because I had breast cancer 10 years ago, and I had Herceptin. Is that an immuno? Okay, so I did have that, but now 10 years later, I have a different breast cancer, and I have immunotherapy this time, so I was just curious why it's like every 3 weeks rather than weekly.
Yeah, I believe the main reason for that is that the drug itself, these antibodies, last a long time, have a long half-life. So they are active for about 3 weeks, and they're slowly declining, and then when they get to a low enough point, you get another dose of them. So I think you're getting coverage of the drug for that whole time.
I think.
Yeah, good luck with your treatment.
Over here.
You mentioned in the middle of your talk, not everyone received the benefit when you did it, the test. It seems to me at the level of the cell, we're all the same. But that's an assumption that must be wrong. Could you explain why it is that you got only 30% or a positive response when you do a test, not 99%?
Yeah, that's a great question. So yeah, why doesn't any therapy work in everybody if it works in anybody?
At the cell level, at the very basic level.
Yeah, so I agree that we all have the same kind of cells at first pass, et cetera. But there's huge differences between us in terms of our basic genetics, like our genomes. We all in this room, the number of T cells we all have circulating in our blood varies by a factor of 4. So some people might have a unit of 1 T cell in their blood, and others might have a unit of 4. I'm a 2.3 myself. And you're very consistent throughout life, and if you're identical twins, they've got similar numbers. So our genetics drives even how many T cells we have, as one example. Then the tumors arise in random processes in each patient. No 2 tumors are the same, so that's another huge degree of variability. And then just the general, how many prior treatments a patient has had, just their general, fitness, as we say, their health, their performance status can vary a lot. So you put all of those factors together, and this is what we find with our CAR T cells that I've been talking about toward the end there. We've treated 95 patients. Some patients respond beautifully, have complete regressions of their tumors. We have patients many years out now who are doing just fantastic. And others, we give them an equivalently made CAR T product, because we follow very strict protocols on how we manufacture these, and their tumor might not respond at all. And to help us solve this, from every patient in our trial, we collect blood samples at up to 15 different time points. And we've got a great bank of blood samples now that we can dig into now and start trying to understand what underlies the what are the determinants of success versus failure of this therapy, and get at that. Is it the CAR T product wasn't as good for this patient? Is it that their tumor didn't have the target on it anymore? Is it something else that was going on with them systemically? So we and the rest of the world are now working on those kind of questions.
Yes.
First of all, thank you so much for dedicating your life to this. Incredible. And is there any toxicity related to immunotherapies like in the past that we do with more traditional ways of treating cancer?
Yeah, thank you for your gratitude and your comment. I appreciate that. And so the question is around toxicities, side effects of immunotherapy. And there are side effects. So the checkpoint inhibitors that I talked about, again, you're taking the brakes off the immune system. So some patients have autoimmune-like symptoms that come with that. A common one is inflammation of the bowel, for example, is a fairly common side effect. Fortunately, those are almost always reversible when the drug is stopped, and they're tolerable and they're reversible. And so patients get through that okay in the vast majority of cases. With CAR T cells, in the early days, there were some quite strong toxicities, just very, very high fever, malaise, achiness, etc. And some patients even need to go to the ICU for respiratory support. A few years into it now, the clinicians have figured out how to anticipate these side effects better, how to detect them early, and how to intervene more rapidly. And so toxicities are much more manageable now. But still, that's an area that we need to keep working on. And I mentioned toward the end, we've got this sort of throttling mechanism that we want to build into CAR T cells now. And we think that'll help a lot, and that you can just, for each patient, just keep an eye on their CAR T cell levels and just get those CAR T cells at the right level, not too high, not too low, just right to get the effect you want.
Just a little follow-up to that. You were talking about high fevers. There was a time when increasing the body temperature seemed to be a desirable thing toward the cancer treatments to work better. Has there been any work in that area?
Yeah, so you're talking about sort of hyperthermic type treatment, and I'm aware of that concept, but I actually don't know much about it, so I won't comment beyond that. But it's the idea of raising a patient's body temperature. Well, I think, for example, while they're getting chemotherapy, as a way to get better perfusion of the drug throughout the body. And I'm not up to date on the latest on that.
There's a question here. Yeah. Question.
So when you're working with the genome and you're working with things that you're looking at the blood and the differences in that, is AI going to be able to help tell what the differences are, and is that being used now?
So this question is AI being used to help us figure out how to make better CAR T cells ultimately, and how to sort of decipher why some patients respond and some don't. So we use AI, the answer is yes, and we use it already in a lot of ways, have for quite a number of years at looking at images of tumors. AI is very good. It's even beating pathologists now in many cases at the ability to look at a tumor and figure out how aggressive it is, and therefore what kind of treatment might be best for the patient. So we use those kind of tools in the research setting every day. In terms of just being able to feed it, we could take all of our data from our 95 patient clinical trial and put it into an AI model and say to AI, okay, you figure it out. What's going on here? And we haven't done that, so I maybe something remarkably great would come out of that. What we do know already though, is that among those 95 patients, there's quite a few different types of leukemia and lymphoma in there. We also know the patients range at different ages. a lot of other factors we're already aware of. And I think, I guess my perhaps skeptical opinion would be the AI might just sort of pick up on things that we already know about. Now, could a sophisticated approach go beyond that? I would welcome that kind of advance. Yeah.
Okay, last question right here.
Okay. I don't know this field at all, so a quick question before my real question. I had read that we only produce T cells when we sleep. Is that just a fallacy?
I've not heard that.
Okay, it's probably
so,
a fallacy. Okay, my real question. I was reading high level about T cells and T cell research, and it appeared that some researchers were moving beyond using mammals as models, and were looking into the ocean and looking at T cell research that was kind of different into fish, vertebrate fish. And I don't know if that's something that CAR T cells have looked at all, or?
Yeah, so the question relates to using fish as experimental models. I assume is what you're getting at for CAR T cell research or other types of character research. So there's a few groups that use fish, and in particular zebrafish, which are aquarium, small sort of aquarium type fish that are apparently transparent, and so they offer some advantages there. You can see into them and see what's going on, and put them under a microscope and see this, that, and the other thing. And there are tumor models you can give them. I'm not aware of anyone going all the way with making CAR T cells, et cetera, against their tumors. So I would say this, that there's probably some things to be learned from fish models, but I do think it makes sense for us to be working with mammals, mice at this point. And then I'm a huge believer in just getting things into the clinic as quickly as we can, because that's when you really learn what works and what doesn't work.
Okay, very last question.
Very hopeful presentation about the science going on, Dr. Nelson. Thank you very much.
Thank you.
In the BC Cancer Agency Research Library, Living Downstream, is a documentary there. Anybody can see it, just go up to the 3rd floor, Lee Street. And Dr. Spinelli and Steinrober say in the documentary that we can't turn the corner on cancer until we have a precautionary principle which will give us the authority to ban all of the suspected and known carcinogens. There's thousands of them. What would be your response to the need for the precautionary principle to guide Health Canada?
Well, okay, I think, so the question is around cancer prevention and ridding our society of known carcinogens, I think is what you're asking about, and I'm all for that. Cancer prevention is still the most important thing any of us can do to reduce our risk of getting cancer. I think you probably heard that about 50% of cancers can be prevented with appropriate precautions. And there are many people out there, it's not my field of research, but people in the epidemiology field who are looking deeper and deeper for environmental carcinogens or other factors that are causing cancer, and then ultimately trying to change regulations to get those out of our society. So I think it's a very important thing. The cure for cancer is not going to be, I don't want to leave you with the impression, I just think CAR T cells are going to take care of everything and we don't have to do anything else. Far from it. We need better prevention, we need better detection, we need immunotherapy, but we need other therapies as well.
Okay, please join me in thanking Dr.